Good morning, dear students. I know you're all enthusiastic to learn more about the cell biology. So, all of you are aware of different shapes of this cell. Dear students, just imagine if our body does not have skeleton, what would have happened? Just a thought, how it would be. So it would be shapeless, like an amoeba would have been irregular. So the same role is being played by cytoskeleton. Myself, today I'll be taking a lecture on cytoskeleton. Myself, Dr. Lakshmi Chandrakant Mushan, Associate Professor, Department of Zoology, DBF Dayanand College of Arts and Science, Solapur. So today we shall be studying about the structure of cytoskeleton and functions played by the cytoskeleton in cell biology. So in 1903, Nikolai Koltsov, he proposed that the shape of the cells is determined by a network of tubules, and he gave the term cytoskeleton for this network. And the cytoskeleton is nothing but a complex dynamic network of interlinking protein filaments which are present in almost all the cytoplasm of all the cells, including the bacteria, archaebacteria. And the cytoskeleton extends from the cell nucleus to the cell membrane and is composed of the same type of proteins like tubulin. We'll be studying about it in detail. So it is the same, the proteins are the same, though the organisms are different. In eukaryotes, cytoskeleton is composed of three main components: microfilaments, intermediate filaments, and microtubules. And when and where required, the cytoskeleton, they are capable of rapid growth or disassembly depending on the cell's requirements. So in the first PPT, here we have seen the imaging techniques. You can see the fluorescent techniques have been used to take the images of the cytoskeleton, and this is the diagrammatic representation of the cytoskeleton, where you can see as the complex network you find the microfilaments, you find the microtubules, intermediate filaments. So three different types, all giving the shape, the internal support to the cell. Thus cell shape is defined because of the cytoskeleton. So coming to the functions of the importance of the cytoskeleton, the primary function of the cell cytoskeleton is to give the cell its shape and mechanical resistance to deformation. Not only that, the cytoskeleton also contracts, therefore deforming the cell and the cell's environment, which allows the cells to migrate. The movement of cells occur because of the cytoskeleton. Cytoskeleton is involved in many cell signaling pathways and in the uptake of extracellular material. During cell division, the microtubules helps in the segregation and pulling of the chromatids towards the poles, thus helping in the segregation of the chromosomes. Not only that, cytoskeleton helps in intracellular transport of vesicles and organelles within the cells. The cell organelles are placed within the cell at that particular position. If you take, suppose, mitochondria, the nucleus, or whatever cell organelle you take, it is in that particular position only because of this network called cytoskeleton. Not only that, in plant cells, the cytoskeleton can be helpful in forming the template for the construction of the cell wall and forming of the daughter nuclei. And in animal cells, especially, the cytoskeleton forms specialized structures like cilia, flagella, spodozoans, lamellipoda, which are helpful in the locomotion of the protozoans. So coming to the structure of the cytoskeleton, the structure, function and dynamic behavior of the cytoskeleton is different depending upon the different organisms, and not only that but also on the different cell types. So whatever it may be, the cytoskeleton, it consists of three components: microtubules, microfilaments and intermediate filaments. You can see here, this is about the microfilaments, where you can see the coil spiral structure, long fiber-like structure, filamentous structure. This is the intermediate filament, and this is about the microtubules. So today we shall just concentrate on the structure of the microtubule and its function. Are you ready? Yes. So microtubules are nothing but, as you have seen in the previous diagram, they are long, hollow, cylindrical and filamentous or fibrillar structures found in almost all the eukaryotic cells, the cytoplasm of all eukaryotic cells, but they are absent in prokaryotes. Microtubules, they are absent in prokaryotes, and they are about 25 nanometer in diameter and 200 nanometer to 25 micrometer in length. And if you see the diagram, the microtubules, if you see the diagram, they're composed of subunits, and these subunits are called as protofilaments, and these protofilaments, they are in a dimer. So the number of protofilaments is the microtubules is variable, and the protofilaments is composed of a series of dimer globular tubulin proteins, and this tubulin is a dimer made up of two similar polypeptides, and the two tubulin dimers are alpha tubulin and beta tubulin. And if you see in the diagram, the two units, they are arranged alternately and in a protofilament, and the tubulin, it shows helical structures with 13 tubulin molecules per turn of the helix. So here you can see the alpha tubulin, beta tubulin forming the dimer, and 13 in a turn, and they are taking a helical turn. You find that the total diameter is 25 nanometers and internally it is 12 to 14 nanometer. You can take down the structure of the microtubules, and these are the different images, the fluorescent images of the microtubules. This you can see the different structures. Now coming to the function of the microtubules. So when you open and observe the cell, you find different shapes, either oval or sometimes it is circular, depending upon different tissue, different cells are, say, epithelial cells or whatever it may be. So this shape of the cell is because of the cytoskeleton. And now, as we have studied just rightly about the structure of the microtubules, now we shall go to the function of the microtubules. So microtubules are the rigid structures which work as a supporting framework, and they give shape to the cell. They maintain the shape of long processes like cilia, flagella, axon of nerve cells, axopodia of protozoa. Here in the previous diagram here, we can see the structure of the cilia. When you take a section of cilia, you see the protofilaments structure in this way. So this helps in the cilia, it helps in the locomotion. And the motion of this cilia in paramecium and flagella in euglena is created just because of the microtubules. Microtubules change the cell shape during cell differentiation. When the undifferentiated cells undergo differentiation, a particular function, a particular shape, this microtubules helps in the changing of the cell shape during cell differentiation. For example, they help in the elongation of the cells in the lens of the eye. The microtubules of many sensory cells that act as transducers which convert the stimulus into the nerve impulse. And not only that, microtubules also help in the transport of cellular materials. And the prime importance or prime function of the microtubules is in the cell division. During cell division in the mitosis, when the chromatids are arranged on the equatorial plate, it is the spindle fibers that develop, that appear, are nothing but the bundles of microtubules which are made of the protein tubulin. These spindle fibers, some are continuous from one pole to the another pole, while some spindle fibers, they are discontinuous, they attach to the chromatids. So by the pulling of these, by the contraction of these spindle fibers only, the chromatids or the chromosomes, they are pulled towards the pole. Thus, the microtubules help in the separation of the chromosomes. Not only that, the centrioles, they are morphologically identical to the basal body of the cilia or the flagella in the animal cells. The microtubules, which are also found in the thrombocytes at the blood platelets of human as well as a rat. And in the plant cells, microtubules help in the formation of cell wall in the plants. So that's all about the cytoskeleton and microtubules. In the next class, we shall study about the remaining ones, that is the microfilaments, which is there in the syllabus. So overall, coming to the summarization, the cytoskeleton is playing an important role in the cell to give its shape, structure. It is present in almost all eukaryotic cells, and it is present in the form of microtubules, microfilaments, or intermediate filaments. Now we have studied about the structure of the microtubules. The microtubules are made of protofilaments, which are composing of a dimer, tubulin units, alpha dimer and beta dimer alternately present. And these microtubules help in the mainly in the spindle formation and shape, in forming the shape of the cells and movement. Thank you.